So good morning everybody, and I will start my class today, and the topic which I will be talking about is on pumps and membranes. It's the last part of my biochemistry portion, and this is something which you people are all aware from your physiology semester CC2, I think, where you had studied about different physiological processes. So in this process especially, where you studied about the urinary system, you had studied and the nerve impulse transmission and synapse. You can find all the pumps and membranes over in these two particular processes. If you read these two particular processes, basically you will have a good idea about all what you need to study. So with this, I'll start sharing my screen. So somebody has come, let me admit that person. Okay, so certain basics about is my whiteboard visible?
Yes, sir.
Okay, fine. So some basics about pumps and membranes. So basically I have pumps and membrane channels. So something about this. On the cell membrane, the plasma membrane, if this is outside and this is inside the cell, outside, ions are unable to pass out or in. So what are the compounds which are easily allowed inside? Any lipid soluble organic compound. It can be a steroid or something. They can easily pass. They can easily pass throughout. Gases, O2, CO2. They can also pass throughout the cell through diffusion. But if you had charged molecules or ions, sodium, potassiums, they don't have a free entry. If you have a water doesn't have a free entry. Water soluble molecules, they don't have free entry. So for entry of all these compounds, you need channels. So a channel basically on a plasma membrane, it allows the passage. This is a channel. And at the same time you have something called a pump. Pump is a device which moves substances against gradient. For example, your the water pump in your house. So from the ground floor to the terrace, this particular pump, it pushes the water up. So what's the function of a pump here? The function of a pump is to push ions against the concentration gradient to the other side. As you know that ions flow in diffusion, it flows from higher concentration to lower concentration, and not from lower concentration to higher concentration. So if you want to push an ion from a lower concentration to a higher concentration side, opposite side, you need to for a pump, and for that you need to spend energy, and that energy is utilized in the form of ATP hydrolysis. This ATP hydrolysis releases energy, and this energy is used to drive ions from from lower concentration to the higher concentration side. The most important among them are the Na plus, ATPase pump. Now this sort of pump is also known as the sodium potassium pump. It is basically an enzyme and it is found in the membrane of all animal cells. Especially it's found in plant cells also, but all membrane of all animal cells. Its property is it uses ATP hydrolysis to drive ions molecules from lower concentration side of membrane to higher concentration side of membrane. So this is the function from low from low to high. This is the function of this pump, and therefore this is a pump for active transport of ions. Most important function which you have seen is that it is useful in maintaining the resting membrane potential of nerve cells. This is the and of course nerve cells, not only nerve cells, of other cells as well. And this effect you know is known as the electrogenic effect, where ATP is hydrolyzed into ADP plus PI and energy is released, and two potassium ions they enter inside and three sodium ions they exit. As you know, in the cell, Na plus inside is low and K plus inside is high. So driving sodium from inside to outside is against the sodium against the concentration gradient, and pushing potassium from outside to inside is also against the concentration gradient going uphill. So this is the essential function. So let us see how it works. Let us take a picture and see how it works. So have a look at this picture on how it works. This is the extracellular fluid on this side. This is extracellular fluid, and inside the cytoplasm, and this is the hydrophobic layer. So under sodium is high outside and potassium is low outside, and inside the cell sodium is low and potassium is high. So potassium cannot exit outside, and it is this channel is closed. Sodium cannot exist and potassium cannot exit, and sodium cannot enter in the concentration gradient. It is closed. Normally it is closed. It doesn't take place like this. But when there is an ATP, first what happens is cytoplasmic sodium, it binds to the the cytoplasmic sodium, it is binding to the membrane, the sodium potassium pump, and sodium binding, once this sodium 3 sodium binds to this membrane here, it stimulates phosphorylation by ATP, and this phosphorylation causes a conformation change. A conformation changes and it flips open in the other direction, and the sodium ions are released outside, and it's expelling sodium to the outside. Similarly, once it is flipped open, 2 potassium also is binding for 2 potassiums on this membrane, and this binds at the same time. This phosphate is released from here. The phosphate is released from here in this figure 4, and then in figure 5, it again, when the phosphate is released, the conformation flips back to this original place. The conformation is over here in this region, and it expels the potassium towards the inside. So this is how the sodium potassium ATP, this is how the sodium potassium ATPase pump works. So let us talk about another pump now, another channel, and that is a very famous channel known as voltage gated channels. Remember your sodium voltage gated channel in nerve impulse. A slight change in the voltage of the membrane because of touching or some stimulus, this channel opens and then sodium enters inside the nerve cell, and then there is depolarization taking place. This particular this happens because of a sodium voltage gated channel. So let us have a look at this voltage gated channel, a picture of a voltage gated channel. Ligand gated channel. So this particular channel, you know, it has got a inactivation gate as well as it has got an activation gate. So at resting membrane potential, the inactivation gate is open and the activation gate is closed, but it is capable of opening. Then from the threshold to peak potential minus 50 volt, you see the channel opens. A slight change in voltage, this channel opens and the activation gate opens, and it enters inside, and then when it has reached plus 30 millivolts, when it has depolarized, when the membrane has depolarized, you can see that the inactivation gate, it closes it, and then sodium cannot enter anymore. Then of course you have the potassium channels opening. That's also voltage. The change in voltage causes potassium, where at plus 30 volts causes the potassium channels to open. That's another thing. So this is how a voltage gated voltage gated channel works. Now let us talk about another channel, and that is your ligand gated channel. Let's have a picture of a ligand gated channel. So this is a ligand gated channel. The ligand gated channel is such a channel. This is the channel. It has got a receptor. This is the receptor. This is the receptor, and this is the same messenger or ligand. This is the messenger or ligand. It binds. Binding causes the channel to open, and this allows ions to pass through. And then when the ligand leaves the receptor, the channel closes, and the ions, they are prevented from entry. So messenger binding causes channels to open, resulting in the entry of ions. When messenger or ligand leaves, messenger release causes channels to close and does not allow entry of ions. This happens in synaptic chemical synapse. During chemical synapse, such a thing can be seen. Let us see a picture of that. So what happens here is that when the synapse takes place, this is a neurotransmitter. These red dots, you can see these are the red dots. These are the neurotransmitters. Red dots are the neurotransmitters. You see these neurotransmitters here. They are released. They bind to the channel. So this neurotransmitter is released. It binds to this channel. The channel opens. Sodium enters and potassium goes out. And then when this neurotransmitter is then the action potential from one neuron, it is passed on to the next neuron through this synaptic transmission. Chemical synaptic. Chemical is the neurotransmitter, like acetylcholine, the red dot. And then binding causes the channel to open. Then sodium from outside, it enters inside, and potassium from inside, it goes outside. This is how essentially this takes place. So let us have a look at the general types of transporters which are normally present. If you read physiology properly, especially the excretory system, urinary system, and the nerve impulse, you'll find most of all the channels and pumps which are present. So let us have a look at what happens, a general idea of what happens in a cell. So just a general cell. Possibly this you can say, call it as some interstitial cells over here. These are the interstitial cells. So this pump you see, this is the Na plus H plus antiporter. You can find them in the proximal convoluted tubules in your nephron. So one sodium enters and one potassium exits. So this is the sodium Na plus H plus antiporter. This is the sodium potassium active transport pump, ATPase pump. At the same time you also have another pump which is present, and this is the sodium bicarbonate symporter. That is, bicarbonate and sodium, they move in one single direction. You have the potassium hydrogen antiporter also present. Apart from them, let us look at the sodium glucose symporters, how they work. So this is the sodium glucose transporter. Sodium enters, it pulls in glucose. Then you have this SGLT, sodium glucose transporter. This is the sodium glucose transporter. This is the uniport, only glucose passes through these channels. And this is of course the active transport, the sodium glucose, sodium potassium ATPase transporter. So with this, basically I covered the whole of pumps and membrane channels, and basically I would recommend you to read about the sodium potassium ATPase pump very thoroughly, as well as voltage gated channel, one voltage gated channel, and you can read also a ligand gated channel. I think these are very important for your exams. So you can read this from your from Wikipedia. I've uploaded certain information in you can see it's in the Google Classroom, and you can read it from your BSc notes about these channels. You can read it from Leninger. You can read it from any standard textbook. You can search on the internet. Lot of information on these channels, but remember to study. You can also listen to my lecture several times, and you can write that down also, and practice these diagrams which you find over here. You can pause them. This will be uploaded on YouTube, so you can pause them and then you can draw these diagrams and you can write it down, so you can already, you have a 4 marks question prepared already. So you can go ahead with this, and I had sent you also the notes on the lipids and also on the molecular biology aspects. If you have any questions, please do feel free to ask me questions. Anybody has got questions? কারোর প্রশ্ন আছে? হ্যালো
স্যার ওই ফার্স্টের যে ছবিটা ওটা 4 নম্বরের ওই পটাশিয়ামটা একটু আরেকবার বলে দেবেন?
হ্যাঁ, বলছি। So what essentially happens is that sodium concentration inside the cell is low, potassium is high, and in outside you have sodium concentration which is high and potassium is low. So cytoplasmic sodium it binds to these slots inside on this membrane channel on this pump. The binding of this it promotes the enzyme ATPase enzyme on this particular pump to be activated, and it hydrolyzes ATP and gets phosphorylated. On phosphorylation the conformation changes and it flips inside out and the sodium is released outside. Similarly, the flipping outside exposes the potassium slots on this particular pump and the potassium binds over here. At the same time binding of potassium it releases the phosphorylation, and the particular enzyme it flips back to its original position of outside in. So the other way round, and once this opens up this potassium is released inside. So because of this phosphorylation of ATP the conformation of this channel changes and sodium potassium are exchanged from one side, from their lower side to the higher side respectively. So this is how the active transport works. দেখতে পেয়েছো?
না স্যার, আমি বুঝতে পেরেছি। আপনার স্ক্রিন শেয়ারিংটা অফ হয়ে গেছে। আমি
অফ
বুঝতে
হয়ে
পেরেছি
গেছিল?
স্যার।
আচ্ছা আচ্ছা, ঠিক আছে, ওকে ওকে, fine, then I end my class over here. Best of luck for your exams. So best of luck for your exams, and if you have
থ্যাংক ইউ।
problems do write to me on WhatsApp or whatever means possible. Feel free to write to me. Okay, thank you very
ওকে
much.
স্যার।
Thank you very much.
থ্যাংক ইউ।